Hey, welcome to the video. We're going to walk through the two different types of fermentation. This is going to happen when oxygen is not present or in enough supply to perform aerobic respiration. So we're going to look at anaerobic respiration, flow diagrams for alcoholic fermentation, and then lactic acid fermentation. So get these into your science notebook. Use this video as a guide if you wish. And let's get started. Even if oxygen is not present, we're still going to go through the process of glycolysis. And this process is simply going to be the breaking or cutting of glucose or of sugar. All right, so we're going to start with the glucose molecule, which is a 6-carbon molecule. And we're going to use a little bit of energy and make it break into two G3P molecules. And then we're going to pull some energy out by converting the two G3Ps into two pyruvate molecules. The energy investment is going to be two high-energy ATP molecules. So two of these ATPs go in, and two ADP come out, and will need to be recycled to ATP. This is the energy investment phase. And then in the energy production phase, we're going to flow in 4 ADP altogether and get a total of 4 high-energy ATP molecules out the other side. We also are able to reduce NAD+, and that happens for each of these G3P molecules being converted to pyruvate. So 2 NAD pluses go in, and we load them up with high-energy electrons, making NADH. All right, so that gets us through to this molecule pyruvate. Now, here is the issue when it comes to anaerobic respiration, right? In order to break down the glucose and move it into pyruvate, and to generate this 4 ATP that we have formed here, we need available to us this NAD plus molecule, right? So if we do not have oxygen available, then the electron transport chain in the mitochondria, along the inner membrane, during aerobic respiration cannot unload their electrons, which means NADH is stuck as NADH. It cannot offload those electrons, and the process backs up. So this is where anaerobic respiration or fermentation processes would enter into the picture, right? So let's take this 2-pyruvate and let's move it through alcoholic fermentation. All right, so one thing we need to keep in mind is that this process of glycolysis, this is going to happen regardless of the fermentation process you go through. Whether it's alcoholic fermentation or lactic acid fermentation, you will always flow through this process from glucose all the way up to pyruvate. You'll get to this guy, right? Now we're going to look at alcoholic fermentation from this point out, and then we'll circle back and do lactic acid. So let's hop on over. All right. So what are we going to do with these a couple pyruvate? Well, we're actually going to convert the 2-pyruvate into 2 molecules of ethyl alcohol, or ethanol. We can mention here that the pyruvate is a 3-carbon molecule, and ethyl alcohol, this is a 2-carbon molecule. So along the way, we've lost a carbon. Where did that go? Well, we're going to be able to pull off from each of the molecules a carbon dioxide. Right? And because we're running 2 pyruvate through here, we will make 2 CO2s. Right? So 2 carbon dioxides will be given off in this process of alcoholic fermentation. And this is why people use yeast for breads and cakes. It will cause your dough to rise. All right? But the more important thing that we do here is that we take a couple of NADHs, and we make use of their high energy electrons to do this, and the result is we free up the NAD+. right? And so this is really kind of like the big deal when it comes to alcoholic fermentation, right? That we can actually take this molecule here and we can cycle it back to the glycolysis steps. Right? We can bring it back up into here and have it used to break down more glucose because it's going to be this end of glycolysis that we get our energy, right? It's going to be this energy production step where we output the ATP. Okay, so this was alcoholic fermentation. What about lactic acid fermentation? Well, to be honest with you, it's really the same beginning step. The only thing we will change is how we end up getting the NAD plus freed up again. Right? So we can simply erase the back half that would be associated with alcoholic fermentation and start from the end of glycolysis on. So we have these two pyruvate molecules ready to be used. We'll zoom back in and we'll walk through lactic acid fermentation. All right, so here we are, we've made pyruvate again. We've generated a net of 2 ATP from the glycolysis step, and now we want to free up some NAD pluses. Right, to do that, we're going to, again, convert pyruvate out, but this time, instead of making a couple of ethyl alcohols, we'll actually make 2 lactate or lactic acid molecules. Now, what you'll notice is lactate is actually still a 3-carbon molecule. So we're not going to give off carbon dioxide here. But what does happen is that in this step to go from pyruvate to lactate, we do take, again, a couple of NADHs, and we're going to oxidize them to where they have lost those 2 high-energy electrons, and we get NAD plus again. Right, so that's really the major difference. Lactic acid fermentation, you produce lactic acid at the end of it. And this lactate that we produce, this stuff here has to actually get cycled back to your liver where it can be broken down into pyruvate and then cycled out to cell respiration when oxygen is available to handle it aerobically. All right, so those are the flow charts when it comes to lactic acid fermentation and alcoholic fermentation. So hopefully you found that helpful.